Yes, so welcome to the day one session on computer online internship and certificate course on computer aided drug and vaccine design. So myself, I am Professor P.K. Praveen Kumar from Department of Biotechnology, Steve Venkateshwara College of Engineering, and I am the Director of Phyto Nanostruct Technologies. Participants, I need a kind of a participant as so kindly mute or I have to remove from the call. Okay, let me go ahead with the today's session. If you have any doubts, okay, you just message in the chat box so that I can able to address you. And now I think I can close my video. Let us start with the presentation. Yeah, up to the participants are nearly the participants are joined from all the backgrounds, UG, PG, PhD, faculty members, everyone have joined in this course. So around the participants or different colleges. Get course is drug design and target identification using pathway databases. So, actually, in these courses, I'll be training you with the PPT initially, and then I'll be coming up with the browser and I show you how to practice with online tools and free open source softwares. That is, I'm going to train only with freewares because that's what you can able to access it. We also have few commercial softwares in our institution. If feasible, I can show you one or two. So more than 90 to 95%, we focus only on the free tools and databases. The participants, major participants are from UG background. They are from second year and third year. So I believe they require basics in these kind of internship and certificate course. Okay, anyhow, I'll be circulating the feedback for each and every 2 days. You can able to provide your valuable feedback, and even if you want to provide uh, a manual feedback, an exclusive feedback, you can provide me through my WhatsApp. I believe you have joined in the WhatsApp group link. So for each and every time, kindly, for once, if you accept our Gmeet invitation, each and every time it automatically log in. Okay, or if you are not accepting the invite, if you decline the invitation, then each and every time you have to admit to join this session. So it's actually a troublesome if the participants are already hearing the class. Okay, and try to join the session before 7 p.m. on every day. And according to the company policies, we'll be providing you the recorded video session that is for each and every day, whatever if you miss, okay, or even if you're not missing anything, if you want to have the content, so I'll be sharing you the video alone, not the lecture notes, because some lecture notes are having copyrights with respect to the results are not yet to be published, so I'm not going to share you the lecture notes. But I will be sharing you some hints like the links, whatever I'm going to teach you. And I also share you some softwares to practice. Okay, the softwares what you have to download and install, and some tutorials. So this can help you to learn. If you want to develop these kind of these tutorials you can develop even from AI tools. Okay, so only thing is that how to learn the skills. That's what most important, and whatever skills we are going to give is related to placement and training. Okay, so many of the pharmaceutical industries where they require artificial intelligence, machine learning, and even the drug design skills, vaccine design skills, they require these kind of things. Okay, so uh, practice and even we try to produce an outcomes in the form of research, in the form of publications, patents, etc. Now let me go ahead with the first slide. So about the phyto nanostruct technologies. So the phyto, and we'll be discussing on the approved courses conducted by MSME. So what exactly is phyto nanostruct technologies and what is the manufacturing division other than the training division? We'll be discussing about that. and we'll be discussing on the types of drug design and something about drug discovery and development, and what do you mean by target and target discovery, how about target identification and targeting convolution, including verification and validation. So I think more about the target verification validation will be discussing on tomorrow. So the basics will be discussing on databases and especially chemical compound databases, protein sequence and structure databases, network pharmacology databases, and few more target identification databases. Now let me come across about company Phyto Nanostruct Technologies. This is a private limited, which is an AICTE and MIC, that is Ministry of Education, Institution Innovation Council, sponsor TPIT and MSME recognized global healthcare startup, which established on December 2024, and we incubated this in SVC Sriperumbudur. Our major purpose is to produce the plant-based precision medicine through novel drug delivery technology, innovative, sustainable solutions. So we are synthesizing and offering drugs as well as nano drugs with of purity above 50%, above 80, and above 95% HPLC pure. And we are also producing above 98% HPLC pure based on the customer's requirement. If you want to procure this Vithaferin A, this is a steroidal lactone compound present in ashwagandha. In genus name is actually Withania somnifera. Yeah, so the popular branded Merck is selling around 1 lakh rupees. Okay, just 10 milligram of the compound expense comes around 1 lakh rupees, but we sell less than that, so around 20,000 for a 98% pure compound. So this is something like we have come up with the process, established process for optimizing the purification of these compounds. Not only we just started with Vitafrini and Vithnodi, now we are having around 26 different phytopharmaceuticals with all levels of purity. And whatever the requirements of the customers, like if the customers are very feel even it's too much price for this 10 milligram compound, then we can also provide in other levels of purity. So only the above 98% comes around that much price, but if you come across above 50%, that's much cheaper than the other purity levels. And whatever phytochemicals we provide, just like Merck, okay, this research chemical grade. That means these phytochemicals can be used for checking the for checking or testing with the cell lines in vitro studies, and we can also check or test on animals, small animals. That's what we call it as in vivo studies. So we can utilize this kind of a compounds or in the form of a nano powder, whatever we provide, and we can able to test for any kind of a diseases. And certificate of analysis, the assurance, we can also provide certificate from IIT Madras. Okay, because the purity levels, even the certificate of analysis is most important to check whether really this is exactly that compound and how about the purity. All these things we will be providing you based on the customers' request. Now let me go ahead with the outcome, like how we got this kind of a startup. This startup was incorporated initially from a DST funded project where I received around 18.3 lakhs, and there I just started working on the effect of the Vithnoid A and Vithaferin A. These are the plant compounds from ashwagandha in a specific target called HSP90 mitochondrial targeting of hepatocellular carcinoma. Actually, this kind of a postdoctoral associateship, this I completed in IIT Madras under the guidance of Professor Michael Kumina. And these are a few of the sponsored projects in 2020 to 2025. My first PhD scholar is Sakti Lalita Sri. She cracked the protein hackathon and she won around 1.07 lakhs in this innovation protein challenge, which is related to this computer aided drug design and also UG students have received a grant of 7,500 for the UG project, so which is also related to in silico studies followed by in vitro studies. And the drug discovery hackathon, this was conducted by AST and CACF, okay, the government bodies. When we face the pandemic situation, they have conducted this kind of an competition, and our students have cracked around 3.6 lakhs under my mentorship. Okay, so actually here we worked on computational studies of discovery of drugs for SARS-CoV-2. Okay, this is the problem statement we have focused, and the students have set it around 3.6 lakhs as the internship stipend. And the phytonano drug technologies originated only from the funding received from AST and Minister MIC, which both are the government of India recognized bodies. Okay, so I have received around 5 lakhs, okay, to establish this kind of a startup for a problem statement. It's not a potent inhibitor. That is, I tried working on computational studies of ethanol A. That is a part of a studies whatever I worked in TST funded project. But here I have used a nanotechnology. Other than mitochondrial targeting technology, I have used nanotechnology. So these two drug delivery technologies fetched me to secure two grants. One is from DST, another from the AST. Out of 1 lakh 5,000 submissions, only 135 innovation secured grants, and only 115 successfully received the grants and worked on that kind of an innovation. So I'm one of the professor in Tamil Nadu. I'm the only professor who has secured this kind of a grant to work on the innovation and to incorporate the kind of a startup. So about the manufacturing division in phytonanos. So we start with the plant and we go with the extract. So I think I can close my WhatsApp, which is disturbing. Meanwhile, I can copy and paste. Okay, copy and paste the links. So the day one links to practice. So I think one faculty has yet to join. 1 minute. I can paste the link to her. Yeah. Now let me go ahead with close the WhatsApp. Yeah, I was explaining you something on. Madam, I think you have joined. Saraswati, madam. Am I audible to you?
Sir, yes, sir. Thank you.
Thank you, madam. I mean, actually, you have to register in the registration form, ma'am. So, if you formally register, we provide you a few more things, okay, to
Sure, sir.
access
Sure,
to.
sir. I'll do it,
Great.
sir.
Thank you. Now, let me go ahead. So, I was presenting something on the manufacturing division in phyto nanostruct technologies. Is this PPT visible to everyone? Kindly tell me. Is this visible? Yes or no?
Yes, sir.
Yeah, thank you, madam. So, because this kind of interaction helps me to know about whether I am continuously connected to the internet or not. So, I request other participants also to interact whenever I request, because I should not uh, disconnect with you at any time. Now, let me go ahead with the manufacturing division. So, this phytonanostuff technologies, we start with the medicinal plant source, can be roots, leaves, etc. We prepare an extract and we purify up to the 4 level of purity, whatever I mentioned initially. And based on the customer's requirement, we formulate with the nano drug solutions. That is, we interact with the different polymers, can be linear or patched chain polymers based on the customer's requirement. And we provide in the form of a nanopolymer. Whatever we uh, we are providing, okay, we also initially validated by checking these kind of compounds, okay, in vitro studies and in vivo studies ourselves. we tried working on this effect of the plant compounds on liver cancer primarily, and then we also worked on breast cancer, lung cancer, glioblastoma, diabetes, inflammatory and autoimmune diseases. So we have already worked on this. So we'll be the we are just submitting the patents. We'll be soon receiving the patents for these kind of application. But we already have nearly five different patents for this kind of a company. So we are selling around 26 different phytochemicals from seven different herbal plants. Okay, so this is about our business model. So a lot of growth opportunity is there, and we are also providing offline internships in SVC. So once in a year, we'll be conducting 5 days kind of a workshops or STTP programs to develop the skills. Okay. Now, these are the certificate courses. Uh, uh, Earlier, I have provided certificate courses to Institute of Innovations, one of the private limited. Now, I have my own kind of a startup, which was sponsored by AICT and MOE. So, this startup, I have conducted nearly seven courses. Okay, seven internship and certificate courses, and I believe this is the third batch of computer-aided truck and vaccine design. So more than 1,000 participants are trained in these kind of courses. So guest lectures delivered. So recently, I mean last month, I have delivered a 1 month internship and certificate course. So this course is limited to computer-aided drug and vaccine design, but a few more additional information I've just trained with respect to artificial intelligence and machine learning training through Python programming. Okay, so this is an advanced course. Whatever you're learning is the basic course. In this basic course, I just going to teach you like how to use the tools and databases for drug and vaccine design. There is also one more course for biomedical engineering students that is related to disease detection using AI and machine learning. Here, in this course, I just deployed two different professors. One is a professor, ex-HOD of a reputed engineering college, and another faculty who is a course instructor. She is actually a founder of a startup of biomedical technologies. Okay, so both are pioneers and they already have secured funded projects in this area. So they worked on images of ECG, EEG, whatever biomedical signaling images we get. Okay, so through that images, how to use the ANML, how to detect the progression of disease. So this is what they train this for 15 days. Okay, so there are a lot of courses we are training related to drug and vaccine design and prognosis, diagnosis, detection, etc. And feedback from participants, we are getting 100% feedback in all the courses. Okay, whatever we have trained, and we are, because it is all helpful for even for training related to placement skills, which is useful for industries. If you come across drug design, the first type of drug design is traditional drug design. So under this traditional drug design, so initially we take sources from plants or from fungi, bacteria. So what if we take, generally, if you take from the plants or animals, we just initially grind it through pestle and mortar, and whatever we have in numerous compounds derived from its source, we use that compounds to check for preclinical studies. Okay, initially we try with in vitro studies and then preclinical studies, but this is not worthy because this is expensive and this won't give a correct kind of a results. And this is the we initially go on the three E's of target as well as ligand. Not to technical concept of one is ligand, okay, or it can be an inhibitor. In pharmaceutical, we call it as agonist and antagonist. Agonist, which can give a desired action, biological action. Antagonist is something which is going to give an undesired action. So generally, this is a small molecule. Whatever I'm telling ligand, is a small molecule. But target is generally a macromolecule. So the most popularly used macromolecule target is nothing but protein. Okay, the proteins are generally used as a target. Okay, and the well-known protein targets or surface receptors. Okay, so that is the receptors present on the surface of the cells majorly act like a targets. Okay, so this is something about terminologies in drug design. So that is ligand is a small molecule, usually a drug, and the target is a macromolecule, usually a protein. In protein-protein interactions, okay, like antigen-antibody interactions, the smallest protein molecule is a ligand. In DNA protein interactions, the DNA is the ligand. RNA protein complex, the RNA is the ligand. Thus, DNA, RNA, nucleotides are small molecules when compared to protein. Okay, so like this, I can come up with various examples. So let me go ahead with this structure-based drug design. So unless we have a 3D structure of both target and ligand, we can't able to minimize the cost. If you want to minimize the cost of checking the drug, okay, coming up with a drug for a particular disease, then we should have this kind of a structure. And so whatever structures we have, okay, so we can able to initially do the computational analysis, and then we can work with in vitro studies by synthesizing the new compounds, and then we can try with preclinical followed by clinical studies. So this is a structure based drug design. Overall about drug discovery and development. So initially we come up with a discovery followed by the development of drugs and testing of drugs. So if you come up to the discovery of drugs, okay, the target discovery usually involves selecting the program. That is, what disease, what is the problem statement we are working. Initially I told our students that the second year students worked on a competition, all India competition called Drug Discovery Hackathon competition, which is conducted in 2020. Okay, what if 41 participants secured internship grants? Out of 41 participants in India, so 6 out of 41 participants are from my, or have received stipend from my mentorship. Actually, this happened they come up with around 30 problem statements for coming up with a drug discovery for SARS-CoV-2. We just selected only one problem statement, and whatever one problem statement we have chosen, okay, so initially we focused on the thing whatever they specified, like they want to come up with a drug for spike glycoprotein. The spike glycoprotein is the most important target for the SARS-CoV-2. So we just targeted the types of spike glycoprotein, like open and closed configurations. So whatever we identified the spike glycoprotein as the target from the disease, initially the program, whatever I mentioned, is nothing but SARS-CoV-2 in our case, and the target whatever they mentioned is a spike glycoprotein. So I've taken the types of spike glycoprotein like open conformation, closed conformation, and the semi-open or semi-closed. Okay, so this is how we have taken these three types of protein targets, and then we have validated the targets. Whether whatever targets we have taken is deposited through wet lab technique like X-ray crystallography or okay, because these are the gold standard techniques where we can get precise structures. If these structures are not available or not deposited through the gold standard techniques, then we model the receptors, where we model the targets. Whatever we model the targets using computational methods, we have to validate it, whether the target quality is good or not. Got it? So this is how whatever we make is a target discovery. And then the heat generation. So in our problem statement of whatever we worked in that discovery hackathon competition, okay, we identified around 1521 drugs, I believe. So whatever 1521 drugs we have taken is from NCBI Pumpkin. So this is actually a free database available in NCBI. Okay, in this database, whatever I told Pumpkin, this is a chemical compound database present in NCBI. Okay, so we have taken that. All these compounds were downloaded in the form of a single data set. And after downloading this compounds in a single file, dataset file, we have undergone all these procedures like virtual screening, asset element. These are the methods actually to identify the best compound from the number of hits whatever we have taken. Okay, the number of compounds we have initially taken is 1521, but initially I want to screen to, I will finally want to screen to a best one compound. For this one best compound, I have gone with the various methods and fixed a kind of a pipeline. Like I use a series of methods, and then in the series of methods in the pipeline, I screen compounds initially from 1521. I screen 400 compounds in one method, that is drug likeness. And in another method, I screened around 100 compounds, base compounds. And in another method, I screened around 10 base compounds. And in another method, I screened 3 base compounds. Finally, out of 3 base compounds, I have to finalize one base compound which can able to target the spike nickel protein. Got it? So, only after identifying the target, then we have to identify the potential lead from the hits. After identifying the lead, if there is a need to go with the lead optimization, it means that if we even based on fragment based screening or some drug engineering, we want to optimize the drug with incorporation or deletion of functional groups. Okay, that's what we call it as lead optimization. So more about this, if you watch the subsequent slides and subsequent session, you can understand that. Okay. So now I'm teaching up to the final year UG and PG, but if you watch subsequent slides, you can understand. Okay, that is after identifying a lead, to optimize the lead, we use some methods and we optimize it. And whatever the best lead we identify, we try to work with in vitro studies, then in vivo studies. In vivo studies is working on small animals like rat, mice, whatever, approved by CCSCA, CCSCA committee. Okay, so each and every institution have an institutional animal ethical committee. They have a biotechnology department and animal approved animal house facility. So if you want to work on the effect of drugs on these animals, okay, initially we must try it with the competition studies, and then if you have a results in vitro studies, that is more valid to get approval from the CCSA committee members to work on these small animals. And after preclinical studies, whatever we work with small animals, we can try with clinical studies, that is the effect of drug on humans. So if you want to check the effect of drug on humans, so which we call the major phases of drug development, okay, so we have nearly phase 1 to phase 4 trials. So to work with humans, there are nearly 4 phases. Okay, phase 1 to phase 4. So related to phase 1, if you want to know about the phase 1 trial, okay, the phase 1 trial is generally working on around 100 healthy individuals. Checking the effect of drug on 100 healthy individuals is what phase 1. If that is phase 1, okay, after check, if it is not giving, if the drug is not giving side effects or adverse effects, then we can go with the phase 2, where we are scaling up the effect of drug in 1000 healthy volunteers. We are scaling up from 100 to 1000. But see, whatever drug we develop, the drug we develop is to check only the diseased volunteers, isn't it? If you are developing a drug for ovarian cancer, then we have to treat the drug only for the ovarian cancer patients. But before trying with our patients, diseased patients, we try with the healthy volunteers. The objective is to check whether it gives any side or adverse effects. So for this, we try with the healthy volunteers. Later, we try with the third phase. If the drug shows successful trials with the phase 1 and phase 2 studies, then we go with the phase 3. So in phase 3, we try with around 1,000, but we try with the unhealthy volunteers, that is diseased volunteers. And then if it shows successful results, then we try with numerous people, okay, who are affected with disease. And also the post marketing surveillance, that is getting our FDA approval. FDA is from US and EMA is from European Medicine Agency. This is from Europe. Okay, so like we have to get permissions from USFDA or EMA, okay, to get approved. Okay, and obviously the post-marketing surveillance approval, everything also includes in phase 4, where the drug is already comes into market and trying with plenty of unhealthy patients. So whatever we discussed initially about the drug discovery and development, we also have similar kind of a terminologies like drug registration. What do you mean by drug registration? And what else further in drug discovery and development? In drug discovery, other than trying in computational studies, that's what we find in rational drug design to structure-based drug design. Okay, so here we can also see the effect or the checking the effect of drugs in vitro biological assays. Like if you want to check the effect of drug in cell lines, for example, if you are working on cancer, then we have to procure the specific cancer cell lines and we have to check the effect of drugs. So all this kind of a screening of drugs also comes under drug discovery. But in drug development, it starts with the preclinical studies to the clinical studies. If you want to work with the preclinical studies to clinical studies, obviously we have to initially, after completing preclinical studies, we have to come up with a pharmaceutical scale up. For example, COVID-19 vaccines like Covishield, Covaxin. Okay, these are the products of purely from India based, isn't it? The indigenous vaccines developed from our nation. So it is targeted 8 billion people. How is it possible? Only because of scale. Can be vaccine or drug. So the scale up is of course most important. And after checking with the ADMET safety characteristics only, we have come across with the formulations. The formulations here, I mean, taking drug in the form of tablets, capsules, or the form of liquid dosage forms like syrups, semi-solid dosage forms like ointments for dermal applications. So all these things, the formulations comes even under drug development. Okay, also comes under drug development and even whatever drugs, okay, in any dosage forms, if you try in humans, can be healthy or unhealthy. So this comes under drug development. And of course, after this only they are trying for the FDA approval. Drug illustration, whatever we want to check the effect of drug on small animals or humans. Initially, we can also check with model organisms like Cyno elegans. You know, whatever disease we get, that disease we can able to induce in the even worms. In roundworms also we can able to incorporate that human disease gene, and then we can check the effect of it. This can also gives you the results whether the drug is treating the disease gene in the worms or not. Whatever disease gene I am telling is the human disease genes, like checking the effect of cancer. The human cancers can also try it with these worms. Not only the worms, we can also try with Drosophila melanogaster. Heard about this? Fruit flies. We can try with the melanogaster. We can also check with the zebrafish. I think uh, the Satyabama Research Park, they are doing plenty of studies on the zebrafish. Not only the Satyabama Research Park, even our institution SVC, we have approved animal house facility, and of course myself is in a the biopharmaceutical scientists. So we are also uh, giving support to the outside institutions to work on research in our institution. Okay, so through consultancies, and actually whatever drug we check, we are not going to check with a single drug. If you want to check with two or three drugs, to check the additive effect, synergistic effect, whatever we try with various forms, okay, like checking bioavailability, checking the efficacy of your drug, all these things comes under tertiary stage, okay, and also working on inheritable genes and working on inheritable diseases. So we try with even in tertiary stage. Now something about target identification. What is target? I told you the target is nothing but a macromolecule, usually a protein. Then if it is a protein, what are the various forms of protein? The major target is protein target is a receptor. The receptor, or whatever I'm telling, is a surface receptor. If you take a cell, whatever uh, receptors present on the surface, this is what I'm calling it as a surface receptor. Other than surface receptor, we can also the surface receptor is generally a structured proteins. These are coming as structured proteins, and which gives a structure the for the cell. Okay, for example, for SARS-CoV-2, we call it as a coronavirus. It's just because the spike-like a protein looks like a corona. I mean, it is like a cap. Okay, so like this, we have so many surface proteins on the surface, like a spike-like protein. But there are also few more proteins present inside the cell. Can be a viral cell or bacterial pathogen. So whatever proteins we present inside is generally classified as a unstructured protein targets. Okay, generally polymerases and proteases. Polymerases and proteases comes as an examples. So if you see the receptors rank number one and the enzymes rank number two, we also find other than the receptors and enzymes, we can also find hormones and ion channel receptors, etc. Now we discuss something on strategies for identifying targets. So there are conventional strategies. The first strategy is analyzing of pathophysiology. Okay, so what are the side effects? I mean, what are the morphologies we are getting for a disease? For example, if a patient is infected with SARS-CoV-2, they have headache, they have the nose block, they find high fever. So these are something about pathophysiology, like whatever the clinicians can detect from if you approach them. Okay, and the next thing is checking the analysis of method of analysis of existing drugs. Initially in the pandemic situation, which happened on March 2019, okay, I mean, at that time, they come up with the, I mean, March 2020, okay, so they come up with the utilization of repurposing drugs. So during that time, okay, most of the clinicians in India and the world, they have prescribed around 8 drugs, 8 antiviral drugs. Whatever 8 antiviral drugs they prescribe, okay, it includes anti-malarial, anti-dengue. Surprisingly, okay, one of the drug they have used is anti-HIV. I don't know how many know about this information. Whether HIV drug can work with SARS-CoV-2, is it possible? But the clinicians don't have any idea. They are not a biotechnologist. They don't know what drug exactly we want to use. They just have an idea we have to repurpose the drug. Whatever antiviral drugs available for treatment of various viruses, they only give that kind of a drugs for treatment of any upcoming viruses or even whatever we found a situation with the SARS-CoV-2. Okay, so we biotechnologists have to think and have to come up with the proper drug discovery. Okay, at that time. So now the new strategy is okay is to find the disease genes and also the disease modifying genes. This is most important because of mitigations you can find the heritable diseases as well as the cancers arise. So we have to find all this, which is most important. And now we discuss something on target deconvolution, target discovery. If you see about the target discovery, this is what generally we are doing. Initially we identify one target. That is, if you take this SARS-CoV-2, the most important target is the spike glycoprotein. After identifying a target, okay, we have to take a series of drugs or series of. Myself, I have taken series of plant compounds. Why I've taken the plant compounds and screened a best plant compound? Because the plant compound has less side effects when compared to the allopathic drugs. Whatever drugs we take in the market or allopathy. So these drugs, most of the drugs are synthetic based. So it can give some side effects also. So to reduce that, I tried with the plant compounds. Whatever plant compounds I tried with 1500 molecules, finally I screened to a single compound. And that's what we call it as one compound, one target in target discovery from a group of compounds. But there is some approach called target deconvolution. In the target deconvolution, whatever drug we have discovered, okay, that particular plant compound, whether this plant compound is going to bind with what type of receptors. Take one compound, try to find multiple targets. Here we take one target, try to find multiple compounds and screen to a single compound. But in target deconvolution, take one compound and find multiple targets. For example, if you take paracetamol, now I think I can interact something because if I take continuously, if you don't know the concept, I don't know how I can make you understand. I can ask you a few questions, kindly interact with me because this is most important to understand about drug design. So if a person is infected with a common fever, okay, is suffering from a common fever, then what is the drug generally a doctor is going to prescribe? Or what drug you would generally take? Can someone answer for this? Unmute and answer.
Paracetamol, sir.
Very good. So whatever you mentioned like paracetamol is the brand name, but the generic name is acetaminophen. If you go to Japan, if you take mention like paracetamol, they won't understand. They have a term like paracet, and in US it is paramol. So we have different brand names. In India, we call it as paracetamol. Okay. So obviously, one should understand that whatever drugs we also have a generic name. Like how I told, ashwagandha is a popular Indian plant, Indian top in 5 Indian herbal plant. But the generic and species name for that is Withania somnifera. Similarly, even for drugs, we have a generic name. Now, my next question is, so how many doses the doctor prescribe for treatment of fever? How many doses per day? This is the next question.
6 hours, 3 times a day.
Very good. Even both answers are right. If you're in the age 10 to 14, okay, we call that as adolescent age, isn't it? The adolescents have rapid metabolism. So just because of rapid metabolism, okay, so these adolescents require 4 doses per day. But practically it is difficult, isn't it? So generally we take morning, afternoon, and night. But 6 hours is more difficult. But we have to take a minimum 3 dosages per day. That's what the doctor prescribed. And uh, the dosage, if we are less than 75 kg, so we are prescribed to take 500 milligram acetaminophen. But if we are above 75, we are about to take 650 mg of acetaminophen. But these kids, They have very less weight. They can also take half of the 500 mg, but they have to take three dosages. This is unavoidable for any age. The elder people or the youngsters, everyone have to take this. If you're not taking three dosages per day, if you take two dosages per day, definitely the drug is not going to cure the disease. It prolongs. Instead of curing in a single day, it prolongs to 3 days, 4 days, 5 days, unless you use the correct dosage. This is not applicable for treatment of common fever, which is infected from pyrogens. Pyrogens, any foreign matter, if it invades. For example, if a patient, if a person got wounded, in that wounded site, you can find the pathogens get entry, and these pathogens can also be killed by our WBCs, white blood cells. These immune cells can attack and kill. But even after attack and killing that bacterial cells, the bacterial proteins, including the membranes, whatever present inside our blood, considered as foreign particles. Okay, both live bacterial pathogens as well as the dead bacterial pathogens, both can induce an increase in temperature in blood. That's what we find a rise in temperature, what we call it as common fever. Okay, so to decide the number of dosages per day and how many dosages and the dosage content, everything we can do it in drug design. Okay, and the final question related to common fever: acetaminophen is the general drug for treatment of common fever, no doubt. What is the target we are targeting in common fever? If you are targeting, what is the common target tissue as well as target protein? Can someone answer for this?
It targets the blood specifically.
Very good. Your answer. May I know your good name? Yeah. Don't want to reveal, no problem. But very good. Your answer is right. We are
Janani,
targeting
sir.
what? Janani. Janani from SVC.
Yes, sir.
Very good. Very good. Your answer is right. So the target is blood. Okay, and actually the target tissue is blood. If I ask you about the target protein, the protein is COX2, that is cyclooxygenase enzyme 2. This is the primary target. You know, we have around 5 to 6 liters of blood. But whatever I take, even 500 mg, even in this 500 mg acetaminophen, we get only 10% of API. API mean active pharmaceutical ingredient. This is the main drug. If you have just 50 mg of the main drug, the remaining 450 mg or binders or excipients like binders. This is what we call it as excipients of the drug. Excipients like other than the API, what are the other molecules present? So that is binders, surfactants, surface tension enhancers, and the we have plenty of things. Okay, viscosity enhancers. Like this, we have plenty of excipients. Okay, our objective is not focusing on developing the excipients, but how to come up with this API. And whatever I'm telling, 50 mg of the major drug, this is targeting 5 liters of blood. Whatever I take the drug, okay, so it is going to cure within 5 to 10 minutes. Within 10 minutes, we'll be getting relief from the fever. If it takes even 15 to 20 minutes, think about if you take an intravenous injection. If you take an injection, paracetamol injection, within 5 minutes, definitely you can find the temperature is reduced because the bioavailability is more in injection when compared to. So what is happening, in blood we have plenty of protein target receptors. We are going to focus on major protein target like COX2. So like this we have to understand. So generally if you go with the screening of drugs, we have random screening. Like we can try to randomly screen whatever we want. But generally in structure based drug design, we go with the non-random screening. There are also drugs discovered accidentally. Okay, the examples are provided. The drug, whatever acetonide, okay, is generally used for treatment of some other disease, but fortunately, it is treating another disease. So, computational approaches for multi-target drug discovery. These were different methods of drug design. More about this drug design, drug discovery, we'll be discussing in subsequent sessions. But I like to give one major idea that in drug design, we have three types of drug design. Number one is the ligand based drug design. Number two is the structure based drug design. There are also, in some textbook, you can also find the third one, that is hybrid based. In ligand based drug design, we need just the 3D structure of ligand. Only the ligand is sufficient. But here we need 3D structure of ligand as well as the receptor. Okay, if you need both the things, then you can able to design the drug for the disease. For hybrid based, it works either like ligand based or structure based. More about this we'll be discussing in the subsequent sessions. So these are some of the major computational terminologies one should learn. Okay, this is something related to drug engineering. So just because I've taken 5 to 10 minutes extra for mentioning about the company and company products, so today I'll be taking session up to 8:10. Okay, I'll be extending my session up to 8:10 so we can see a few more hands on. Now let me come across this drug engineering. Okay, sometimes the drug molecule is not a single molecule. So whatever I tell API, that can also be two molecules. For example, in my DST funded project, I worked on a strategy called mitochondrial targeting strategy. You know, in cells, we have cytoplasm, we have so many organelles, and one of the powerful organelle is the mitochondria. The mitochondria is a double layered membrane. Targeting this mitochondria is very difficult. So, if you have a mitochondrial targeting drug, and if you have a specific drug which can able to cure the disease, we can link it. That's what. For linking this, we use linker. Okay, for linking drug 1 and drug 2. Similarly, you can also fuse the drugs. You can also use the other strategy like merging the drugs, or sometimes we can use a cleavable link. Like after reaching the site of action, the drug is going to target. Generally, if you take a drug alone, the drug will be metabolized in liver when instead of targeting a desired tissue. So instead of metabolizing much in the liver, if you want to target something like kidney cancer, if you want to target the kidney in kidney cancer, then obviously we have to use a cleavable linker where using the photocleavable linkage through variation in pH. That is, after going to a particular pH, this linker is going to cleave, and so the drug is going to target only the desired organ. Drug development is a team sport. If you see pharmaceutical industry, we can find formulation scientists, clinical development scientists, clinical operations specialists, toxicologist, regulatory affairs specialist, safety physician. Of course, clinical pharmacologist is most important. Who can link you? Okay, there are much more terms to find inside this. We'll be discussing in subsequent sessions. Now let me directly go to few tools. Okay, the first tools, I take you to the browser and explain whatever present in the PPT. Instead of showing that, I directly take you to that. The first website I'm going to explain is NCBI PubChem. So instead of PubChem, I just go to the NCBI. And the URL of the NCBI is ncba.nlm.nih.gov. This is a US government website. Okay, so this NCBI, National Center for Biotechnology Information, is coming under the National Institute of Health, which is a major body, a funding body from US. If you see in this NCBI, we have plenty of databases. So the most important databases what we use on today is the PubChem. Here, if I want to download a 1D, 2D, or 3D structure of a drug or any chemical compound, in my studies, I try to use a plant compound called phytopthrin A. So if I type phytopthrin A, you are getting so many results, around 34 results I got. Not only got phytopthrin A, but also got derivatives of phytopthrin. So many derivatives we can find. Okay, so we find around 34 hits. And if you want to get this information, I just get inside by clicking this hyperlink. You know, so not only we can find the physicochemical properties of the particular plant compound, but we can also find the vendors who can distribute this, who can able to sell, and we can get the structures information, etc. Now, if you see what are the information I can find, structures, chemical and physical properties, okay, and then you can find the spectral information, chemical vendors, interaction and pathways, patterns, literature. So all these things, okay. If you are a layman and if you want to know more on this, on working on a particular compound in research, then first you have to watch this NCBA part. So for example, if I click this chemical and physical properties, you can find this molecular weight, partition coefficient, donor count. How to download the structure information, the structure information is you can able to download from here, okay. The 2D structure. In the structures, there is something called structures. If you click that, it take you to the 2D structure. So the popular downloadable coordinates in 2D structure is SDF, structure data form. Okay, so now I'm going to download this compound. So I'm going to create a folder in desktop called CADVD 26. So this is the folder I'm creating. Now whatever compound I'm downloading is a Vitafrin A 2D structure, isn't it? I'm going to rename it like this. Like this, I'm also going to download 3D coordinates in STF. Now, whatever I'm going to download these 3D coordinates, you can able to see how the 2D varies with 3D in the structure. So after downloading this compound, I am going to open this in a visualizer. The visualizer, what I'm going to show you is BioVia Discovery Studio Visualizer, which is a freeware. So I suggest all the participants to install this Discovery Studio 2023 visualizer, which is a freeware. From the first session to the last session, we need this visualizer. So to open this molecule, if I get double click, I will be getting like this. Later, right click, show in graphics view in left click. After right click, left click. So whatever you see, this is the 2D structure. Now I'm going to open a 3D structure in Discovery Studio Visualizer. So again, you are clicking show in graphics. Now see the difference. What is the difference you can find? If I click the display style to the white background, you have an idea. So now I replaced with the white background. Even 2D structure, I'm going to replace with the white background. Go to the graphics, background color, white, apply white. See, whatever 2D structure I found, this is in a single plane. But here, whatever I can find, see, it's in multiple planes. So it is flexible here. It's not in a single plane like a 2D. This is how we can find a variation of 2D to 3D. It's a single plane, but multiple planes. So for docking simulation, we have to use only 3D. So we should not download 2D for any drug design applications. Now let me go ahead with the next thing. The first thing, whatever I showed you, is the NCBI. Okay. Then what are the other most important databases that I can show you? So, like however I search Vitafrene, you can type any drug, aspirin. Okay, you'll be getting the aspirin drug. So nearly 141 results required. Other than PubChem, if you want to use the structure, the structure, is a molecular modeling database. The name of structure database present in NCBA is molecular modeling, MMDB. In the structure, for example, if I want to download an insulin protein, I type the name of the term as a keyword, name of the protein as a keyword. Simply click search. If I click search, we'll be getting 1762 results. If you want to get the mutant insulin, you can download the specific mutant, or else you can search for the specific insulin. Not only you can find insulin from Homo sapiens, but we are also finding from different species. Suscrofa means pig, swine. So this is a swine insulin. So if you see 60 years before, where the recombinant technology is not established, so we are the patients, the diabetic patients are taking insulin only from the samples, especially from swine. So this is the structure of the insulin. Okay. So for example, if I want to open the structure of this wine insulin, let me go to this structure. To simply click download in this PDB ID. So now I'm going to save this. So if you see this PDB ID, this is a 4-letter alphanumeric letter. Okay, you can able to see here as a download to download this protein structure, and the identifier what we use is the alphanumeric letter. This structure was discovered in 1997, but this is updated in this database only in 2017. Earlier then it was deposited only in PDB database. PDB is another 3D structure protein database. Later in MMDB it was deposited, and this was discovered from experimental method called X-ray diffraction, and the resolution is this. Okay, now let me go ahead with the next database called PDB. What is PDB database? So PDB is an exclusive database, okay, for 3D structures of proteins. So we got a PDB ID like 1ZNI, isn't it? Can we type the same 1ZNI? Either type insulin or type the specific PDB ID. If you see, the total number of structures available is around 1.2 lakhs, 1.25 lakhs. So out of 1.25 lakhs, one structure is 1ZNI. So we can find the same information. Okay, so as I mentioned, this is deposited in 1997 released here. Okay, and the X-ray diffraction. So more about this information you can get. But for downloading the files in PDB format, you can use legacy PDB format. Click the download files and go to the legacy PDB format. You can download the same similar file like whatever you download from in. The next database I'm going to show you is the TrugPack. So in Google, simply type the TrugPack. So if you are the initial users, okay, the first time you are going to try with this, you have to register. You have to register. The URL is go.trugpack.com. So after getting inside, okay, that is after registration, you have a login and password, get into login credentials, type in it. Now here I typed aspirin. Now if I type acetaminophen and click search, this is a drug database. Not only can find the information of drug molecules, but also phytopharmaceuticals information we can get from this drug pack. The phytopharmaceuticals generally used as drugs. Okay, so that covers also available. So now I just typed acetaminophen. We can find all this information: pharmacology, interactions, chemical identifier, spectra, targets, enzymes, etc. So if you have time, just explore on these most important databases. Okay, so register and login. The next thing, whatever we are going to try, is the KAGT. The KAGT initially was free, but now it is blocked actually. Okay, it is blocked, and we can able to see just the API alone. The KETT is better known for downloading, getting the information on pathways, genomes, drugs, targets. This is the most important database. Okay. So now, last month, I believe they have blocked using this website. But of course, this is the most important thing. But what else we can able to access other than this? So you can able to find one database called StringDB. This is a protein-protein interaction database. So you can see the protein-protein interactions are very important for precision medicine. So you can find PPI interactions from 12,535 organisms, and you can find around 20 billion interactions. Am I audible? Yes? Yes?
Yes, sir.
I can able to complete within 3 minutes. Thank you. So I was explaining something on the string database. So whatever is unexplored, tomorrow I'll be taking you directly to the hands-on. Okay, much of hands-on. Okay, now let me take you to the string database. Simply I click search. You got this kind of a window. In my case, I worked on plenty of targets. The most important targets I got most is the trap one. The trap one is the name of a protein, also called as heat shock protein subtype. If I simply click trap one and I click search, it takes you to the so many species of trap one proteins. Like as I mentioned, it is a heat shock protein subtype. Okay, so default species of Homo sapiens. Simply click continue. So it takes you to the Homo sapiens heat shock protein subtype. So whatever molecule, whatever protein exploring, you can able to see the top 10 proteins interacting with this. So this is the protein trap, and this is interacting with 10 different proteins. If I want to increase the number of interactions to top 100, simply go to the settings. Instead of the 50, 10 interactors, I use the 100 value here. The maximum interactors is 100. Click update. Now you can see the top 100 proteins interacting with the trap1 protein. So our trap1 is located here at present. Like string DB, we also have one more database called Stitch DB. The Stitch DB distinguishes with string DB. Not only we have PPI interactions, but we also have drug protein interactions. Drug protein interactions. So the drug protein interactions you can able to see from this example, like rofipoxib. This is a inhibitor for COX-2. Okay, so this drug is binding with few proteins. If you want to know more about this protein, click this info. You'll get the protein information. The protein is prostaglandin endoperoxide synthase 2. So like this, you can see the drug interacting with proteins. We also find protein-protein interactions. Other than this, you can find this kind of information. So like this, I have shown you plenty of databases. Okay, and for in each and every day, every session, for the first 5 to 10 minutes, I recap what we discussed in the earlier session. So I'll be conducting you the questions in the form of poll, or I'll be asking the questions in the chat box you have to answer for. The interaction gives much more better understanding of what we discussed. Okay, so from tomorrow onwards, we'll be interacting with more kind of questions and answers. Okay, so these are some of the website links to be watched. You can watch the links, and whatever is uncovered, we can see it on tomorrow. Okay, any doubts at present, you can ask. You can stay back and ask. Any doubts? I've completed today's session. Anything you can ask. Anything. There's nothing called silly.
Sir, may I?
Yeah, yeah. Please.
Sir, in drug deconvolution and target deconvolution and target discovery, what I understood was, I'm just asking if it is right or not. What I understood was, in target deconvolution, we have one compound which interacts with so many targets, whereas in target discovery, we have so many compounds which can interact. We are finding which compound interacts best with the one target which we are looking for. Is it right, sir?
You are right, madam. For if you want to work with the target deconvolution, if you go with the Swiss target prediction, you go with the Swiss target prediction, which is available in Swiss target prediction.ch, which I'll be teaching you in the coming sessions. Okay, so here they ask, it asks you to paste only the smiles format. Smiles format is the 1D format we can get from even pumpkin. If you see the PubChem, okay, we have earlier I told you now how to download 2D and 3D. The same database, but in a PubChem, okay, if you search, we have smiles format. Okay, simply go to the specific compound and take the smiles format. Not only from the PubChem, we also have plenty of databases like DrugBank, CHEMBL, CHEBI. So okay, so if you type smiles, you get like this. This is a canonical smiles. Simply copy and paste it in the smiles box. Okay, so if you search predict targets, it gives numerous targets where it exactly can find. Okay, this is what target deconvolution. Okay, I think I have cleared it out, but target discovery, you have to even work with similar kind, then you can understand. Okay,
Yes, sir.
that we use explicit tools for it. Okay, any other doubts from any other participants? Good. Okay, thank you for the interaction. From the beginning onwards, you have interacted well, which made me more interesting to teach. Okay, we have special care to teach more concepts from the next session onwards. So join the session before 7 p.m. Okay, so I sharply start at 7 p.m. Try to close at 8 p.m. So thank you. Good night to everyone. Bye to
Thank you,
everyone.
sir. Thank you, sir.
Thank you.